Hello students. In this video class of 30 minutes duration, we will be discussing the reactions of monosaccharides. Now, basically, the reactions of monosaccharides are the reactions of the subgroups or the components of monosaccharides, the keto group or aldehyde group or hydroxyl alcohol group, or some of the formations like acetal formation or ketal formation. And sometimes this forms the basis of detection of these molecules also, of these sugars also. And not all the reactions of monosaccharides have been included. Some reactions like formation of disaccharides or polymers, or some other reactions like glucose forming the precursor of ascorbic acid, for example. These have not been included in this class. Now, reactions of monosaccharides. Now we will be discussing enediol formation, reduction. Reduction means reduction of monosaccharides. Ozone formation, oxidation of monosaccharides. Reducing property. Reducing property means reduction by monosaccharides. Furfural formation, glycoside formation, ester formation, and amino sugar formation. So these will be dealt in this class. Let us see one by one. Now, first of all, enediol formation. Action of alkali sugars dilute alkali with monosaccharides, which are having free functional group in them. All the monosaccharides are having free functional groups in them. They undergo intermolecular rearrangements to form enediols. The enediol structure is shown here. The hydroxyl groups, two hydroxyl groups, are connected to carbon, which in turn are connected through a double bond. So this is the enediol part, and this is an enediol structure, 1,2 enediol form. And I can note here glucose and fructose, which can interconvert through enediol formation, which can get interconverted through enediol formation. One of the things that you have to note here is that fructose and other ketoses with the keto group in them, they are not having reducing property, unlike aldoses. The aldehyde group is the functional group which imparts reducing property to monosaccharides like glucose or ribose or galactose or mannose. So it is unlike keto group. But here fructose and other ketoses can get converted to aldose through this enediol form. So that is the significance of enediol formation here. It actually reducing property is given by given or acquired with the help of this interconversion through enediol forms. And glucose, fructose and mannose are having the same enediol forms, because the first the difference between them is around the first and second carbon, that is the anomeric carbon and the adjacent carbon. And this is through tautomerization that they get interconverted. Now, tautomerization means it's a simple relocation of hydrogen atoms. Relocation of hydrogen atoms. In glucose and fructose and mannose, a shift of hydrogen atom gives rise to tautomers. This reaction is also called Lowry-de-Bruyn-Alberda von Eckenstein reaction. So that's a pretty simple name to write as well as to pronounce. And this is how glucose, mannose and fructose, this is the shift of hydrogen atom. You can note here when we compare the actual structure, it's just a shift of the hydrogen atom, as you can note. Let us take the case of fructose. This is C double bond O in fructose actually, and this hydrogen was actually on the first carbon, CH2OH. So this is a shift of single hydrogen atom only. Now we move on to reduction to alcohol. Reduction of monosaccharides to alcohol. With the strong reducing agents, aldoses give corresponding alcohol like glycerol from glyceraldehyde, ribitol from ribose, sorbitol from glucose. Like that it goes. So this is alditol. If an aldose is converted to alcohol, it is alditol primarily or generally. Here, D-glyceraldehyde in the presence of sodium borohydride. It's not a very strong reducing agent, but in the case of monosaccharides, it acts very quickly it acts as a reducing agent. So glyceraldehyde gets converted to its alditol form, namely glycerol. And glycerol is a structure which we know. CH2OH, CHOH, CH2OH, as well as glucose. D-glucose gets converted to D-sorbitol, its alditol form. That's not about the aldose alone. Ketosis also get reduced to its alcohol, corresponding alcohols. One difference is that when ketones are reduced, the carbonyl carbon becomes asymmetric, like happens here. C double bond O becomes asymmetric like this. HOH on the right, OH on the right side, HOH, OH on the left side, so that it can become or can give rise to a mixture of two alcohols, like what happens here, D-sorbitol and D-mannitol, which are derived from reduction of D-fructose. Now, sugar alcohols are natural products, and two of the sugar alcohols written here, xylitol and ribitol, they are actually used as non-sugar sweeteners also. And we move on to another property of monosaccharides, osazone formation, another reaction of exhibited by monosaccharides, osazone formation. Now, sugars react, or monosaccharides react with 3 molecules of phenylhydrazine. And that means excess of phenylhydrazine, let me say, while we do practicals, to give hydrazone and then osazone. So this is actually a 3-step process. The first step is shown here. This is phenylhydrazine. With loss of water molecule, it becomes phenylhydrazone. So this is the structure. The first carbon is rearranged. Now, the second step, addition of one more molecule of phenylhydrazine shown here, and with the loss of an ammonia and an aniline molecule, it becomes this step. This is the second step. You can see the second carbon has been rearranged, and one more molecule of phenylhydrazine shown here, that also actually reacts with the second carbon to become this structure. So it's around the first and second carbon that the changes of the reactions go on or take place. This is with the release of water molecule, becomes osazone. So this is glucose, becomes osazone. This is fructose, becomes osazone. And you can note here both the osazones are having the same structure, which means the first and second carbon, the change is in first and second carbon in the case of glucose, fructose, and also mannose also. This is a single step representation for osazone reaction, 3 molecules back to back, it becomes like this. So this is the first and second carbons which have changed their structure, and yeah, this has already been discussed. The first and second carbons, glucose, fructose, and mannose are having the same crystal structure for osazone, crystallization structure, and which can be under microscope, which can be observed under the low power of microscope, and can observe it like this. For glucose, fructose, and mannose, the same kind of crystals. It is described as needle-shaped crystal or broom like crystal, and this is for lactosazone, a disaccharide actually. It gives powder puff or tennis ball shaped crystals under low power of microscope, and this is sunflower petal shaped crystals of maltose, another disaccharide. Here again, this is a more clear picture of fructosazone. Fructose and mannose and glucose are having same structure, and this is for sunflower maltosazone and powder puff lactosazone. This is the drawn structures. Same. Now, oxidation of monosaccharides. Monosaccharides on oxidation give 3 different series of compounds, which depends on the type of oxidation and the type of condition under which oxidation is carried out, and basically it depends on the type of oxidizing agents also. And as a result, 3 products, one is aldonic acid, under another condition uronic acid, and saccharic acid are formed. First of all, let us see how aldonic acid is formed. Now, with mild oxidizing agents like bromine water or alkaline iodine, we get what is called glyconic acid with glucose. So glucose under mild oxidizing agents or oxidizing condition, it gives gluconic acid, and galactose under mild oxidizing agents, it gives galactonic acid. You can see the first carbon here, hydrobromous acid is used. The first carbon, the anomeric carbon, the carbon in which the functional group was that was oxidized, and carboxylic acid group is formed, gluconic acid. Now, the second condition in which the first carbon, the anomeric carbon, is protected, the aldehyde group is protected by suitable methods, and strong oxidizing agents are used. So what happens is that the sixth carbon, that is oxidized, and what happens here is that a carboxylic acid group is formed. This is more of an enzymatic method usually followed for this type of oxidation. So glucose gives glucuronic acid, and galactose gives galacturonic acid. Now this is oxidation under strong oxidizing agents like concentrated HNO3 and heat. Under this condition, glucose gives glucosaccharic acid. So what happens here is that the first carbon, both the first carbon and the sixth carbon, both of them get carboxylated to give glucosaccharic acid. So what happens to galactose? For galactose under the same conditions, galactose gets converted to galactosaccharic acid. This is also called mucic acid, as shown here. Now, glucose can also be oxidized using glucose oxidase enzyme, a particular enzyme, which is a method usually used for assessment of glucose in biological fluids. So glucose oxidase converts glucose to gluconic acid and H2O2, and in this method, actually the concentration of the production of H2O2 is assessed so as to assess the concentration of glucose in the given sample. So this is what is done. Principle of glucose oxidase method. So this is the first reaction, gluconic acid and H2O2. This H2O2 is to be assessed, and another enzyme, peroxidase enzyme, is used so as to release nascent oxygen, and the nascent oxygen released is assessed using colored dyes, which when oxidized, it gets it shows some certain colors like pink color or so, and depending on the concentration of or intensity of that color, the concentration of H2O2 as well as the concentration of glucose can be assessed. So this is more of a laboratory method used to assess glucose concentration in biological fluids. And now we can discuss the reducing property exhibited by monosaccharides and most of the disaccharides also, except for disaccharides like sucrose or trehalose, which are not having free functional groups in them. Generally all sugars with the free functional groups in them act as strong reducing agents, and they can reduce like Ag2+, Hg2+, or copper2+, etc. Now what we use is that dilute alkali containing compounds which can release these ions, Ag2+ or Hg2+ or copper2+, are used. And since we are using dilute alkali, first of all, the monosaccharide will turn to enediols. So enediol formation will occur, as we have learned in previous slides. Sugar can be converted to its enediol structure in dilute alkaline medium. Now dilute alkali also does another thing, because if we are using compounds like cupric hydroxide, the alkali ionize cupric hydroxide to give cupric ions or to release cupric ions like this. So this is cupric hydroxide. Under alkaline conditions, it releases cupric ions. Now the next step is cupric ion react with enediols. So enediol structure, the enediol gets oxidized to give sugar acids. Now obviously enediol, the carbon in enediol, carboxylic acid groups are formed in enediol structure. So this becomes sugar acid, as you have learned again in the previous slides. So we know what sugar acids are. And what happens correspondingly, this cupric ion gets reduced to cuprous ions. And the cuprous ion combine with hydroxyl ions to form cuprous hydroxide. Cuprous hydroxide is having a yellow to orange color, which on further heating turns to brick red colored cuprous oxide, like this. So this is cuprous hydroxide formed. Then this is cuprous oxide, brick red color. Now this is why in for example in Benedict's reaction we can see a play of colors. So that is from blue to the reagent color changes to from blue to green to yellow to orange to red. So this is the basis of Benedict's reaction also. Benedict's reagent actually contains sodium carbonate, copper sulfate is here, which can release cupric and then cuprous ions, and it also contains sodium citrate. In alkaline medium, sugar form enediol, and this is the reaction which follows. And finally, sugar acid also is formed by oxidation of sugar. This is the reaction that happens. Sugar enediol, it oxidates to sugar acid, and correspondingly this also happens. Copper sulfate releases cupric ions, then gets reduced to cuprous ions, then like this, copper hydroxide and cuprous oxide. Now the significance is reducing action of sugar on cupric ions is utilized for identification and quantitative estimation of reducing sugars in biological fluids. Now apart from Benedict's test, this reducing action is also employed in Beaufort's test as well as Filling's test. So this is the reducing action exhibited by sugars. Now furfural formation of monosaccharides upon action by acids. With dilute acids, aldoses generally they are stable, whereas ketoses are not stable with dilute acids. They give furfural forms. With the concentrated acids, even aldoses give furfural form with the loss of 3 molecules of water. So this is basically dehydration, loss of 3 molecules of water. Now pentoses give furfural forms and hexoses give hydroxy methyl furfural. So in general, ketoses are more susceptible to the action of acids. And the significance of this reaction is that the furfural derivative, it can condense with organic phenols to give colored condensation product. You can see how. So this is a ribose. Upon here we have used concentrated HCl, 3 molecules of water are gone. You can see formation of double bond here. This is the furfural form of ribose, and orsinol is used here. This is an example for organic phenol, orsinol, and gives blue-green colored condensation product. Again, this is glucose. Now glucose, here we are using concentrated H2SO4. Again, 3 molecules of water are gone, double bonds are formed, and hydroxymethyl furfural is the structure formed here. Here we are using another type of organic phenol, namely alpha naphthol, to give purple or violet colored condensation product. Now pentoses give this furfural form, hexoses give this furfural form. So this is basically 5-membered ring structure. So this is what is shown here is 5-membered ring structure. So tetrosis or triosis will not give furfural form or this color reactions. So this is the basis of Molish test shown here. Just one of the preliminary test to identification for the identification of carbohydrates. You take alpha naphthol with sugar solution, shake it, and add concentrated sulfuric acid through the sides of the test tube, and you can see the purple colored ring here. So this is Molish test. And apart from Molish test, for the identification or quantification of pentoses and hexoses, we use rapid furfural test, Selinov's test, and Bayes orsinol test also. So all these tests, the basis is furfural formation. And triosis and tetrosis obviously will not undergo such reactions, and they cannot be detected using this particular type of tests, or for example Molish test. Now, glycoside formation by monosaccharides. The hydroxyl group that is attached to the anomeric carbon atom of carbohydrates in solution, it shows an unusual reactivity, and the derivatives called glycosides can be formed from this OH group that is attached to the anomeric carbon. So this OH group reacts with alcohols, so OH groups again, to form glycosides. For example, here CH2OH, ethanol is involved, methanol is CH2OH. Methanol is involved in glycoside formation here from D-glucose. The product is methyl D-glucopyranoside. That's a glycoside, and the other examples are also there, digoxin, fluorescein, etc. Now, these are chiefly produced by plants, and some of them are toxic also, especially to the cattle that feed on them. Cyanogenic glycosides are very toxic, deadly poison, but in small doses they act medicinal also. They act, for example, as muscle relaxant. And cardiac glycosides are used in treatment of cardiac dysfunctions. So they have got some medicinal uses also. Now we move on to the ester formation. Here hydroxyl groups can be esterified. Hydroxyl groups in monosaccharides can be esterified with acid groups to give compounds like acetates, phosphates, benzoates, and so on. And among these, phosphates are better known to us because they are we constantly meet phosphates in during metabolic reactions. So one of them is here glucose 1-phosphate, which is called Cori ester. Another one is glucose 6-phosphate called Robinson ester. Glucose 1-phosphate, obviously the first carbon, or the anomeric carbon, OH group, is reacting with a phosphate group to give glucose 1-phosphate, whereas glucose 6-phosphate is formed by a reaction of the 6th carbon CH2OH, that hydroxyl group, with the phosphate group. And there are plenty of such esters which we see in metabolic reactions. One of the metabolic steps, namely HMP pathway, has got a number of esters. For example, glucose 6-phosphate, ribulose 5-phosphate, ribose 5-phosphate, cellulose 5-phosphate, cedoheptulose 7-phosphate, and so on. So that is ester formation. And finally, amino sugar formation of monosaccharides. Now, amino sugars are formed by actually hydroxyl groups of glucose. Here we are again taking the example of glucose here. Hydroxyl group of glucose is substituted by amino group. And mostly this happens on the 2nd carbon of glucose, as shown here. And acetylglucosamine as well as glucosamine are formed by substitution of the 2nd OH group with the amide or amino groups. Actually, more than 60 sugars are known, with one of the most abundant being N-acetyl D-glucosamine, which is the main component of chitin. Now, chitin is the 2nd most abundant polysaccharide in nature, which comes behind cellulose only, and chitin is a long chain polymer of N-acetyl glucosamine. Now, another example is peptidoglycan, which is a major structural polymer in most bacterial cell walls, and it consists of glycan chains of repeating N-acetyl glucosamine and N-acetyl muramic acid. Both of them are amino sugars, and these residues are cross-linked with the peptide side chains to form peptidoglycan. So with that, we come to the end of this class. Thank you.